I am François Bertucci. I am MD, PhD. I am co-director of the Laboratory of Predictive Oncology at CRCM, co-director with Dr. Émilie Mamsier. I am also a medical doctor in medical oncology, and at IPC, I manage patients with sarcomas, and I work in the field of precision medicine, which has a link with what we do in the lab as a translational research lab. Our lab has been involved for many years in the genomics field, and we were involved in the first studies which consisted in the profiling of breast cancers to identify signature, prognostic, or predictive of response to treatment. And we have continued to work with these technologies, and now our lab is involved in 3 axes. One is a profiling of tumors from patients treated at in situ polycalmate at the DNA, RNA level to identify signatures. The second axis is to develop preclinical models, such as organoids or PDX or cell lines, to deepen the results that we observed on the clinical samples, and to try to better understand the role of such alteration in the disease or in the resistance of treatment. The third axis is complementary to the 2 previous axes. It consists in the development of new drugs that we could use for our patients. It's a systemic treatment based on antibody drug conjugates. And in the lab, we have an engineer, Marc Lopez, who identified 10 years ago, perhaps, a new antigen target, which is expressed at the surface of the tumor cells and not in the normal cells, which is Nectin-4. And based on our results, we develop an antibody drug conjugate anti-Nectin-4 in a startup that Marc co-founded with Daniel Olive. And this startup is Emergence Therapeutics and is going to launch next year a clinical trial in which IPC will participate, of course. The second part around ADCs is the identification of new ADC targets. And another important part is to try to better understand the mechanisms of resistance to ADC. It's very important for us because, of course, we work on the same campus, but we don't have many occasions to be together, to discuss together between the different teams of the CRCM and of the Department of Translational Research. And such a meeting is very useful to launch collaborations, to better know other people who work beside us, but that we don't see every time. Yes, well, very happy to be here. I am very happy to be here for this anniversary of the 50 years of CRCM. We have seen a very big revolution during the last years, from the beginning of translational research to now with new technologies which are more and more complex, which gives us access to the actual complexity of the tumor cells, of the cells, at a level which is unprecedented, because we have access now at the single cell level, but also within the cell to different compartments of the cell. So it's very, very exciting, but that gives us an idea of the complexity of the disease and of the tasks that we have to face.